Good
Yes,
morning.
ma'am.
Good
Morning,
morning,
ma'am.
ma'am. Morning.
How are you, ma'am?
I am fine. So can we start the class or shall we wait
Yes,
for
ma'am.
others?
Start.
Yes, ma'am.
We'll start.
Sorry?
Yes,
Start.
start. Start.
So today we discuss about that structure of bacterial cell. Structure of bacterial cell and maybe someone would like to start with the structure. And there is a question, what variant of cell structure in bacterial cell? Can you tell me?
What, ma'am?
Ma'am.
What variant of cell structure in bacteria?
Yes, ma'am. Structures outside the cell wall like capsule, flagella.
No.
Yes, ma'am.
Which type of cell structure is having bacteria?
Ma'am, basically cell wall, but only one bacteria which is called mycoplasma. It is bounded by cell membrane. Otherwise, all bacteria are covered by the cell wall.
No. Type of cell structure, I'm asking. Type of cell structure.
Ma'am,
See?
Coccus bacillus, phylum.
No. It is just morphology. Да? I am asking you about structure of this that type of structure of cell. And in nature there are two types of cell structure are known.
Yes, ma'am. Gram positive, gram negative.
No. Listen to me. Eukaryotic and prokaryotic. So eukaryotes, all that higher plants, animals, the human cells, some protozoan cells, these are all, they all have eukaryotic cell structure, and you should know about it from biology and histology, right? And prokaryotes are all bacteria. Bacterial cells have prokaryotic cell structure. And there is another question. Can you tell me what is difference between eukaryotes and prokaryotes?
Ma'am, prokaryotes mean before the nucleus.
Sorry, once again.
Ma'am, prokaryotes mean that organism lack a nucleus, and eukaryotes have the nucleus.
Yes, absolutely right. So eukaryotes have no nuclear membrane. That's why they have no clearly identified and separated nucleus in the cytoplasm. In prokaryotes, this is called the nucleoid. It's not nucleus, it is called nucleoid in prokaryotic cell. In eukaryotes, it is nucleus. In prokaryotes, it is nucleoid. That was a main basic point of dividing all cells into two groups. What other differences do you know between eukaryotes and prokaryotes?
Ma'am, eukaryotic cells contain organelles like mitochondria, lysosomes, and such other
Endoplasmic reticulum,
Yes.
endoplasmic reticulum,
Endoplasmic
Golgi
reticulum.
apparatus,
Golgi apparatus, yes, ma'am.
which
But,
are absent in
prokaryotes.
prokaryotes, yes.
Yes,
But
ma'am.
prokaryotes.
Yes.
No, ma'am, ribosomes are present in bacteria.
But, ma'am,
But
the
different, the
small
70s, the
unit. Yes,
Yes,
ma'am.
ma'am.
And in eukaryotes, 80s. And, ma'am, also the prokaryotes have a external cell wall which is made up of a peptidoglycan.
Yes, this is a component of cell which is not found anywhere else.
And ma'am, in eukaryotes, only parasites have motility, but in prokaryotes, the bacteria have the motility.
I'm sorry. Here, there is my. Today came my grandbaby and not giving me chance to talk to you all the time. I'm sorry. Okay, let's continue. So difference between eukaryotic and prokaryotic cells that they mentioned, and in microbiology, we will only talk and discuss about the prokaryotes, only about the prokaryotes. So I'm sorry, it's about your program making and syllabus and etc. And we are discussing about it.
Okay,
Your
ma'am.
university is
Okay, ma'am,
going
no
to
problem.
get accreditation of, that's why.
Inshallah, ma'am.
Yeah, we are working on the program.
Ma'am, how much chances we will get soon accreditation?
Sorry?
How much chances we will get accreditation soon?
No, I don't know. It seems to be next week will come that commission, maybe. I don't know what they're saying about it, and we are now preparing the documents for this. And
Ma'am, our university is prepared.
we are preparing my subject. I am doing my subject. Each teacher doing their own subjects. And other documents, I don't know. It's up to your university.
You are PhD doctor.
Sorry?
You are PhD doctor.
Yes.
Ma'am,
I am
on your
associate professor, PhD.
ma'am, on your slides written as a Kyrgyz professor of Kyrgyz State Medical
Yes,
Academy.
I am. My main place of working is the Kyrgyz State Medical Academy.
Ma'am,
Okay.
can you tell what are the main differences between the Kyrgyz State Medical Academy and Adam University?
No, Adam University is very young, just now made it. It's 3 years only, as I understood. Kyrgyz State Medical University is 80 years already.
But ma'am, Adam University director said it is working on medical faculty 2003 local faculty.
No. Mainly people study in medical academy to get medical education.
Okay, ma'am.
Your Adam University, I don't know. I heard only last year that such university we have in Kyrgyz Republic. Okay. No, it is a private, you see. This is a state academy. Your university is a private university.
Yes, ma'am. Yes.
Yeah, that's a difference. Of course, it is a big difference between state and private. Okay. So we were talking about structure, right? So prokaryotic cell structure. Prokaryotic cell structure containing of 3 parts, appendages, cell membrane, and cytoplasm. That But here you may see, you may observe, yeah? Structure of bacteria, yeah? There is there are essential structures, yeah? Like cell wall, cell membrane, cytoplasm, nuclear material, and non-essential structures. These are capsule, flagella, pili, spores. These are non-essential. Are you listening me? Hello.
Yes, ma'am. Yes, yes.
Okay. Okay. So this at first we should start from discussion from the appendages. Yeah? Appendages, these are external organelles like flagella and bacterial flagella and pili, pili or fimbria. Okay, can you tell me about the flagella?
May I, ma'am?
What is your name?
Abdul Moit, ma'am.
Abdul what?
Moit.
Just a moment, I should find you. Abdul Muit. Okay. Okay, please, flagella and pili.
Okay, ma'am. Ma'am, it is a long white-like appendages, which is the subunits of single proteins. It is intervened chains. They have energy for movement. It is provided by adenosine triphosphate. Its motive force is provided by the adenine triphosphate. They have the characteristic number and location of flagella. Some bacteria have one. This flagella is located at one end, and some bacteria have the flagella all around. It only certain bacteria have flagella. Many rows have, but most cocci do not have flagella. But spirochete have flagella like structure and this called axial filament. Flagella have two important reasons. Some species of motile bacteria, for example, E. coli, are common cause of urinary tract infection. They play important role in pathogenesis by propelling the bacteria up the ureter into bladder. And some species of bacteria identified in the clinical laboratory for the use of specific antibodies. And pili, it is the hair-like filaments that extend all over the cell surface. They are closed and shorter and straighter, and they are composed of subunits of pili proteins in helical strands. It is mainly found in the gram-negative bacteria, and they have two important roles. Uh, there is a
What is the
special
function of flagella?
flagella, ma'am, it is some flagella for function is movement. For example, E. coli from urethra to the urinary bladder, and in some flagella like salmonella, they have used for the laboratory identification for the antibiotic.
Then?
And in the pili have the function.
Just a moment. So, main function of flagella is to move, giving the ability to move the bacteria,
Yes.
right? Movement, and it is a filamentous, filament. You see that filament, and it has a hook, system of rings, which is combined to the bacterial cell cytoplasma membrane. And what in electron micrograph you may see flagella like that. You see that long filamentous, and distribution of flagella may be different. Look at this. Peritrix, Lophotrix, Monotrax, and Amphitrix. Different, but most of pathogenic bacteria have peritrichous flagella. Okay, what can you tell me how to check out in the laboratory, how to check out the motility of bacteria?
Ma'am, with the help of flagellar proteins.
No, mortality. For example, you cultured bacteria, right? For example, you have grown bacteria and you have to check out either it's motile or non-motile. But how can we do it?
No, ma'am. I didn't clear it.
Ma'am, can I tell?
You can, what is your name?
Saad Ahmed.
Saad Ahmad, okay.
Ma'am, by the use of specific antibodies against flagellar proteins.
Yeah, this we can detect the antigenic structure of
Yes,
bacteria.
ma'am.
Antigenic
Okay,
structure.
ma'am.
But what about mortality itself?
Ma'am, mortality is due to the flagellar proteins.
No. Checking the, observing the mortality. We can observe it. What? Look at this detection of bacterial mortality. Да? Mortality. We can use flagellar stain. Mortality test medium can be used. When we inoculate the bacterial mortality test medium, да, it can swim in semi-solid medium and can give the growth not in the place where you inoculated. It can spread through all that medium and tube and grow in that place. With direct microscopic observation, in of living bacteria in a wet mount. This is the main method of detection of mortality. This means a hanging drop test, which is widely used in laboratory. Okay? Hello?
Okay, ma'am.
Yes, ma'am.
Understand?
Okay, ma'am.
Okay, what Abdul Muid, please,
Yes,
could you
ma'am.
continue with the pili, fimbria or
Okay.
pili?
Ma'am, it is the hair-like structure that extends from the cell surface. They are shorter and straight, and they are composed of a protein arrangement in a helical strands. They are mostly found in the gram-negative area. They have two important role. Special kind of pilus like sex pilus forms the attachment between the donor and the receptant bacteria. During
Recipient and
conjugation.
just a moment, the sex pili is playing role in exchanging of the exchange of genetic information between two bacterial cells, and it is called conjugation process.
Yes, ma'am.
And common pili?
And in a common ply, the attachment of the bacteria to the specific host human cell to produce the necessary infections, initiation of infection for some organisms and mutants of Neisseria, gonorrhea that do not form pili. They are non-pathogens.
And
Yes.
so this presence of pili is showing the bacterial virulence. That means if pili present, it is means that bacteria is virulent. Okay. Thank you. Cell membrane. Maybe someone would like to answer cell membrane. The cell membrane contains capsule, cell wall, and plasma membrane. You should start from the capsule. Maybe someone would like to answer about it.
May I?
Yes, ma'am. Can I start?
Okay. What is your name?
Saad Ahmed, ma'am.
Saad Ahmad. Okay. Capsule. Capsule.
Yes, ma'am.
Please.
Ma'am, capsule is a gelatinous layer covering the entire bacterium. Ma'am, it is basically composed of a polysaccharide, which is a lipid, and ma'am,
This is a capsule.
Polysaccharide vary from species to species. Ma'am, for example, there are 91 different types of Streptococcus pneumoniae. Ma'am, it is a ma'am, capsule is basically the determinant of virulence of many bacteria, since it limits the ability of phagocytic ability of the bacteria. Ma'am, there are negative charges which are present on the polysaccharide. The negatively charged cell membrane of the neutrophils prevent it from ingesting the bacteria. It means, ma'am, capsules act like a anti-phagocytic. It does not allow the bacteria to engulf. And ma'am, specific identification of an organism can be made by using antiserum against the capsular polysaccharide. Yes, ma'am, that's it.
Mhm, mhm. So, what is main function of
Ma'am,
that
main
capsule?
function of capsule that it's like an anti-phagocytic.
Mhm, mhm. And what other functions of capsule do you know? Antiphagocytic means we can say not only antiphagocytic, protection of bacteria
Yes, ma'am,
from
protection.
host immune defense.
Yes,
It
ma'am,
helps
yes, yes,
bacteria from
Okay, host immune defense. Then
yes.
what other?
Ma'am, also opsonization.
Opsonization is doing by antibodies, then?
Yes, ma'am, antibodies, which enhances the phagocytosis of bacteria.
No, we are talking about capsules. We are not
Yes, yes,
talking about phagocytosis. Huh?
yes, yes,
So
ma'am.
this capsule also protecting from drying, drying desiccation.
Ma'am, interaction with other organism,
Mhm.
Yes,
Drying
ma'am.
desiccation,
Also used in
permeability,
several vaccines.
permeability barrier as a defense mechanism against pathogen,
Mhm.
phagocytosis, and other nutrient cell.
Mhm. How to detect the capsule? Ahmad, Saad Ahmed. Detection of the capsule.
Ma'am, capsules are
Hello?
detected by a yes, yes, ma'am. Ma'am, this is a test which is called Quellen reaction.
Uh-huh. It
Yes,
is
ma'am.
serological method, right?
Yes,
It is
ma'am. Yes,
serological method.
yes,
Mhm.
ma'am. Yes, in which, ma'am, capsular polysaccharides are used as the antigens in certain vaccines because they are capable of eliciting protective antibodies. Ma'am,
Mhm.
like Staphylococcus pneumoniae, Neisseria meningitis, and these bacteria are used in such reactions.
Mhm. So capsule
This is
quelling
basically,
test.
ma'am, swelling phenomena.
Uh-huh.
Yes, ma'am. Swelling phenomena. This is basically.
No, swelling occurs if capsular antigen and antibodies in the serum are specific to each other. They will combine with each other and swelling occurs. If they are not specific to each other,
Yeah,
swelling will not occur.
okay, ma'am.
That's why it's called a serological reaction. But without serological reaction, what other methods do you know?
Ma'am, other methods. Ma'am, by using the anti-serum against the
No,
capsular polysaccharide.
no. You already mentioned about it. Other method, it is staining, demonstration of capsule. What look at this.
Okay, ma'am. Okay. Staining.
Negative staining by India ink or neurosin stain. Capsule appears as a clear halo around the bacteria, whereas both of the bacteria and the background appear black.
Ma'am,
We
but
use
Nigeria.
India
Okay.
ink staining for demonstration of the capsule, and you may observe, you may see here that background is black, bacterium also got the color of India ink. Our surrounding part, which is colorless, this is a capsule.
Yes, ma'am.
Here also, this is a complex staining. This is simple staining. This is a complex staining. When we stain use this staining, and additionally we stain with safranin. Because of safranin, bacteria became red color. You see, capsule anyhow remaining colorless around that bacteria. Understand?
Yes, ma'am.
You wanted to say? You were saying something. But about what?
Ma'am, I say that Nigeria is gram negative. Yeah.
And
Ma'am, then method for identification we use gram negative bacterium. Gram negative.
I don't understand you.
Ma-
Okay, Neisseria is gram negative.
Then, ma'am, we use what staining process? Gram negative
Gram
staining or
staining, of course. Gram staining. It is showing that it is a coffee bean shaped gram negative bacteria. That means it is maybe Neisseria meningitis or Neisseria gonorrhea, but we have to use other methods for its identification.
But ma'am, Staphylococcus is gram positive cocci. Then we also use gram staining.
We gram staining, please do remember, gram staining we use to all bacteria.
Okay, ma'am.
All bacteria.
Okay,
It is important feature of bacteria. Important and important method of classification of bacteria is a gram positive and gram negative. Okay.
Okay, ma'am.
Okay. So cell wall. Saad Ahmed, please.
Ma'am.
I am listening.
Yes, ma'am. Cell wall is the outermost component common to all bacteria, but ma'am, only one bacteria, which is mycoplasma, is bounded by the cell membrane, not a cell wall.
Yes.
And ma'am, these cell wall is present externally, like such as and external to the cell wall, appendages are present, like we already discussed about flagella, capsule, and villi. And ma'am, cell wall is located external to the cytoplasmic membrane, and it is also composed of peptidoglycan. And ma'am, peptidoglycan, it provides the structural support and also the functional support and maintains the shape of the cell. That's it, ma'am, about cell wall.
No, why? That's it. Many functions cell wall have. What other functions of cell wall do you know? You see that what's the difference between gram positive and gram negative bacteria cell wall structure? What are the differences you can see? That peptidoglycan layer very thick in gram positive. Here very thin.
Yes, and the teichoic acids are present in gram positive and absent in gram negative. And also the endotoxin, which is lipopolysaccharide, is absent in gram positive and present in gram negative bacteria.
Yes. Absolutely right. Okay, and what other functions? It gives shape, permanent shape. That's why we can classify bacteria as a rod shape, spiral shape, and cocci shape, etc. Okay, what other functions do you know? Confusing.
Yes, ma'am.
Well, these are it is essential, essential, and provide ligands to adhere and receptor sites for drugs or viruses, for symptoms of disease. What you mentioned, endotoxin, will cause the symptoms of disease, provide for immunological distinction and immunological variation among strains of bacteria, and they can form in certain situations, environmental conditions, they produce spheroplast. These are three variants.
Yes, ma'am.
Cell wall deficient forms. L form, protoplast, and spheroplast. Here you may observe these three types of bacteria which are lacking of cell wall. Normally they have cell wall, but in some when they affect, for example, by penicillin or lysozyme enzyme, they are forming L forms. That they can lose the cell wall, and these L forms. They are named as the L forms because first they were discovered in the Lister Institute. Yeah? And this bacteria, L forms, they can multiply, they can grow, yeah? Without cell wall. Even they are lacking of cell wall, they can grow, they can multiply and produce disease. You know, and the L forms, it is not forever. For example, as soon as you stop using penicillin, cell wall may appear again. Is it clear for you?
Yes, ma'am. Yes, yes.
The L formation means that phenotypical adaptation for the environmental stresses. And it is a phenotypical mechanism of antibiotic resistance formation.
Yes, ma'am. Okay, understand. Ma'am, which gene produces the resistance against penicillin?
Against of penicillin producing resistance is plasmids, mainly plasmids, penplasmid, which is giving bacteria ability to produce enzyme, penicillinase enzyme, which is called as a beta-lactamase enzymes.
Okay,
These
ma'am.
enzymes destroy penicillin.
Okay, ma'am. Suppose if
Penicillin not attacking the bacteria. Bacteria, by producing beta-lactamase enzymes, are destroying penicillin.
okay, okay. Ma'am, suppose if our body is resistant against penicillin, then what the next treatment? Suppose penicillin doesn't
Once
show.
again,
Ma'am, suppose penicillin, our body is resistant against penicillin. Penicillin doesn't show any effect on our body. Then what is next? What next treatment?
not your body. It is not affecting bacteria. Please do not mix.
Ma'am, yes,
Penicillin is not affecting bacteria because of such resistance formation. L forms or either penicillinase enzyme production, it doesn't matter.
Yes.
And you have to choose the antibiotic with another mechanism of action. For example, you can choose antibiotics of group of quinolones.
yes, ma'am.
Yeah.
Primaquine, chloroquine.
Yeah.
Am I right?
Another mechanism of action. Do you know?
Yes, ma'am.
Because why? No, in more detailed information about this,
Yes.
you will get in the topic of antibiotics. You have separate topic of antibiotics, and we will discuss about it.
Okay, ma'am. Okay.
Since you see that antibiotics are all using and targeted to affect essential parts of the bacterial cell, like cell wall, plasma membrane, ribosomes, for example, synthesis of proteins of ribosomes, and etc. No, this is we will discuss, as I told you, more detailed in the topic of antibiotics. Don't worry, this topic
Okay,
you have.
okay, ma'am.
Okay,
Okay, ma'am.
and then
Okay, okay.
more the information you will get.
Okay, ma'am.
Okay. Next component of bacterial cell, it is plasma membrane. Please, Ahmed Saad, could you complete this cell membrane? Plasma membrane. The plasma membrane is lying between cell wall and
Yes,
cytoplasm. It's
cytoplasm.
separating cell wall and cytoplasm. Okay,
Yes,
could you
ma'am.
explain?
Ma'am, plasma membrane is also made up of peptidoglycan. Ma'am,
Plasma
peptidoglycan.
membrane? No, not made up of peptide. No.
Ma'am, plasma membrane of bacterial cell wall.
Da.
Ma'am,
ये तो लिखा ही नहीं बाबू
मैम
लिखा हुआ है यार. Ma'am, in bacteria, ma'am, plasma membrane is present between the cell wall and the cytoplasm. Ma'am, it is basically consists of a lipid bilayer.
Yeah.
And ma'am, it is semi-permeable membrane. It means it selectively permeable membrane. And ma'am, it regulates the transport of materials.
Who is talking? Saad Ahmed?
Yes, ma'am, Saad Ahmed.
Okay.
And ma'am, plasma membrane regulates the transport of materials entering, and it controls the entering and exiting from the cells. And also, ma'am, the functions of plasma membrane, like it acts as a barrier. And plasma membrane is a selectively permeable membrane.
Yeah,
And
you can see that.
Yes, ma'am.
Osmotic or permeability barrier, transport system location, energy
Yes, ma'am.
generating function, synthesis of membrane lipids, then murine, which is
Yes, ma'am.
peptidoglycan,
Murin protein. Yes, ma'am.
assembly and secretion of extra cytoplasmic proteins, coordination of DNA, chemotaxis, location of specialized enzyme. So another
साहब मैम को कहना था वो तो बुक में लिखा ही नहीं हुआ
अब बाद में बताऊंगा.
Okay, you see that this mesosomes, so
Yes, ma'am.
can you read this?
Yes, ma'am. Ma'am,
Please read it.
ma'am, whole slide. Just
May I,
mesosome.
ma'am?
Can you see the slide?
Yes,
Yes,
ma'am.
ma'am.
Okay, you can read it.
Yes, ma'am. The plasma membrane of prokaryotes may invaginate into the cytoplasm or form stacks or vesicles attached to the inner membrane surface. These structures are sometimes referred to as mesosomes. Such internal membrane systems may be analogous to the cresta of mitochondria or the thylakoids of chloroplast, which increase the surface area of membranes to which enzymes are bound for specific enzymatic actions. Mesosomes may also represent specialized membrane regions involved in DNA replication and segregation, cell wall synthesis, or increased enzymatic activity.
Understand? Mesosomes are, this is invagination of plasma membrane inside of the cytoplasm. And mesosomes involved in DNA replication and segregation and cell division. What function of mesosomes? And also respiration, also enzymes for respiration also located in this mesosomes and plasma membrane. Okay. And spore formation also. You see, plasma membrane participates in spore formation also. You know, plasma membrane, you should understand that plasma membrane is very important component of bacterial cell, essential and important. Okay.
Yes, ma'am.
Okay. Saad Ahmed.
Yes, ma'am.
Okay, who is next? Cytoplasm, please.
May I, ma'am?
Tell me your name. I don't see you.
Abdul, ma'am.
Just a moment. Two?
No, ma'am. One.
Can I, ma'am?
Abdul, your name is what?
Abdul.
Abdul Muit, you already answered, enough?
Yes,
Yes,
ma'am.
ma'am.
I want another person. You should
may
get
I?
some marks.
Okay, ma'am. Sorry, ma'am.
Ma'am,
Yes,
may
ma'am.
I?
Can I, ma'am?
Just a moment. Who?
Mehtab Hassan, ma'am.
Who?
Mahtab
Mahtab
Hassan.
Hassan. Okay, Mahtab
Yes, ma'am.
Hassan. Mahtab
Yes, ma'am.
Hassan.
Cytoplasm is a gelatinous liquid that fills the inside of a cell. Uh, it is composed of water, salts, and various organic molecules. Some intercellular organelles, such the nucleus and mitochondria, are enclosed by membrane that separate them from the
From
cytoplasm.
mitochondria. They have no mitochondria.
No, ma'am. Enclosed by membrane that separate them from the cytoplasm.
So you tell me, what is um, cytoplasm? Да? What you see the cytoplasm. Да?
Yes, ma'am.
Cytoplasm is a colloidal, да? Colloidal structure on which are fixed that structural components of the cytoplasm, да, of the bacterial cell. Да? These are ribosomes. Да?
Yes, ma'am.
What other organoids do you know? Ribosomes,
Ribosome,
nucleoids,
nucleus, vesicles, rough endoplasmic reticulum.
plasmids, да, genome.
Yes, ma'am. Golgi apparatus.
Golgi
Cytos.
apparatus not present in bacteria. Forget about eukaryotes. Totally, you should forget about the eukaryotic cells in microbiology. We are talking about prokaryotes. Understand?
Yes, ma'am.
We are talking only about prokaryotes, and please, you should only talk about this. Golgi apparatus, mitochondria, it's all about eukaryotes. What prokaryotes may have? You see, in the beginning, I mentioned that eukaryotes are simple in the structure, simple than eukaryotes. Prokaryotes are simple than eukaryotes. You know, more simple. Well, you see that they only have nucleoid, ribosomes, lysozyme, inclusions, bodies, plasmids. You see, plasmids.
Yes.
The ribosomes are 70S in size. Okay. Mahtab Hassan, please,
Yes.
can you tell me about the what is the function of nucleoid?
Sorry, ma'am.
Function of nucleoid. Nucleoid function. Nucleoid function.
Is essential for controlling the activity of the cell and reproduction.
So nucleoid is keeping the genetic information, the genetic information of the bacterial cell, and transferring genetic information from one generation to another generation, to the new generation. This is an important function of that nucleoid. What about plasmids? What can you say about plasmids? Plasmids.
Ma'am, its function? Yes, ma'am. Plasmids are used in genetic engineering to amplifying to or produce many copies of certain genes.
No, I'm not saying about in genetic engineering. For bacterial cell, what is role of plasmids for bacterial cell?
Ma'am, it may defending the host cell by producing toxin. Some plasmids facilitate the process of replication in bacteria. They may contain genes that enhance the survival of an organism,
Yes, yes, yes. So
other by
plasmids.
killing organism by defending the host
So
cell
you
by
see,
protein.
you see that here written that prokaryotes sometimes possess smaller extra chromosomal pieces of DNA called plasmids. Né?
Yes.
So that means plasmids are lying separately from the nucleoid, né? And carrying additional. You see, the key word is additional genetic information for bacteria. Né? And presence of plasmids may give some advantages to the bacterial cell in surviving. Yes, you are right, in surviving, and means to produce resistance to external factors. No? Especially it is antibiotics. Да? What Han Saad Ahmed, do you remember I told you that plasmids, pen plasmid, penicillinase enzyme production plasmid. So this is about what this plasmid. I was telling you that these plasmids nowadays became main mechanism of antibiotic resistance formation and development. You know, that's why these plasmids give, as I told you, advantages to bacterial cell. And there are, according to the type of genetic information which can carry the plasmids, they are classified into R plasmid, resistance formation, PN plasmid, fertility plasmids, F degradative plasmids, and etc. Variants of plasmids. And in genetics, we will have a topic of genetics, and in genetics we will discuss about it. Okay, in more details we will discuss about the plasmids. Understand?
Yes, ma'am.
Okay, please tell me about ribosomes. This is Mahtab Hassan. Mahtab Hassan. Mahtab Hassan. Where are you?
Yes, ma'am.
Please. Continue, please. Ribosomes, you should complete your answer this
Yes,
time.
ma'am. Bacterial ribosome are composed of two subunits in which 50S and 30S are composed to 60S and 40S in eukaryotic cell. Ribosomes function as a workbench for protein synthesis, whereby they receive and translate genetic instruction from the formation of specific protein. Yes, ma'am.
So, main function of plasmid is to synthesize protein. Да?
Yes, ma'am.
Okay. Next, inclusions, please.
Yes, ma'am. Ribosomal subunit are composed of ribosomal RNA and protein. Ribosomal subunits with different S value are composed of different molecule of ribosomal RNA as well as different proteins.
Inclusions, bacterial. Не хочешь, что ли? Yeah. Inclusions in bacteria. What inclusions? You see? Often called the cytoplasm, or often contained in the cytoplasm of prokaryotic cell. Yeah?
Yes.
But read please this slide.
Often contained in the cytoplasm of prokaryotic cell is one or another of some type of inclusion granules. Inclusion are distinct granules that may occupy a substantial part of the cytoplasm. Inclusion granules are usually reserved material of some sort. Many bacteria accumulate granules of polyphosphate which can be used in the synthesis of ATP. These granules are termed Velatin granules or
The chromatic granules. Да? They are characteristic features of
Term Velatin granules are metachromatic granules. They are characteristic features of the cyanobacteria. They can be settled.
Okay, so what does it mean, inclusions? Inclusions are what? Storage, inclusions are storage of nutrients, and inclusions usually no chemical compositions, maybe like polyphosphate, metachromatic granules, and they can be used to synthesize the ATP. That means during when bacterial cells living in an environment where nutrients are a lot, they accumulate some amount of that nutrients inside that their cells, which are known as the inclusions, that nutrients, they storage nutrients. And when, for example, after some time they get the situation of malnutrition, they start using these nutrients, which they storage during having enough nutrients, and now they are using it as a when there is a deficit of nutrients. Okay, please continue. Mahtab Hassan.
Yes, ma'am. Yes, ma'am.
Lysozyme function. Lysozyme function, these are group of enzymes which are participating in the digestion of nutrients. Lysozyme. So you see bacterial cells containing nucleus, plasmids, volatin granules, ribosomes, and lysozyme group of enzymes. Okay. So we discussed about the cell structure today. If you have any questions, I am ready to answer.
No, ma'am.
No questions? How many? How many people are present now?
So, 6, ma'am.
3.
Ma'am, 7.
Okay. 1, 2,
Maybe by
3, 4, 5.
Oh, by
6
30 को
people.
भी count.
6 people are present. Why? Some people are left the class.
Ma'am, they
no,
have
ma'am.
some.
They have internet problem.
Do you know when I have classes, my classes in with the medical academy students, they never have internet problem. But they are also living in Pakistan, same as you are.
No, ma'am. But some people are living in the backward areas, so they have internet problems.
Maybe you think that students who are studying in medical academy living only in cities? I think
Yes,
they also
ma'am.
live in
Yes,
some
ma'am.
village also. They live in the villages also. But they never have a problem with the internet.
But अगली class भी
Okay. Okay. Just a moment. Just a moment. Sorry. Just a moment. Wait. I forgot to give you. Can you see it in chat? Look at the chat.
No,
Yes,
ma'am.
ma'am.
Yes.
Your next topic. Aseptic, antiseptic, sterilization, disinfection, nutritional bacterium, culture media, classification and preparation of simple broth and agar. So this is your
Okay,
topic.
ma'am.
And today your lecture is cancelled.
Oh, thanks, ma'am.
Do you know about it?
No,
No,
ma'am.
no.
You tell us.
Okay. And because I cannot do it. And your lecture will be on Wednesday, and on Wednesday I will try to give you this topic. in lecture.
Inshallah,
Okay,
ma'am.
ma'am.
But
Okay, ma'am.
anyhow, this topic you may find in your books also.
Ma'am,
Okay,
which
ma'am.
chapter?
Okay.
You will find physiology of bacteria
मैम को
chapter.
चैप्टर का किधर पता यार.
Physiology.
Okay,
we are
ma'am. We will try to
moving
find.
to physiology. Yeah?
Yes,
See,
yes, ma'am.
you start studying of physiology of bacteria.
Okay,
Okay?
okay, ma'am. Okay, okay.
Okay. Thank you. See you.
Paracho,
Goodbye.
ma'am.
Goodbye.